MADHURA WALVEKAR and I have chosen the research paper titled Machine Learning Enhanced Extraction of Biomarkers for High-Grade Serous Ovarian Cancer from Proteomics Data. So in the first slide there is introduction and this slide tells us that it introduces the research focus on discovering new biomarkers for high-grade serous ovarian carcinoma. The primary goal is to improve the diagnosis and prognosis of HGSOC by identifying biomarkers that are more accurate than those currently used in clinical settings. In this study, it leverages machine learning techniques applied to publicly available proteomics data from ovarian tissue and serum samples to achieve this goal. In slide 2, the research motivation in this paper is given. In this paper, the motivation behind this paper is that it stems from the limitations of current biomarkers like CA125 and HE4, which are not always sensitive or specific enough for accurate diagnosis. This slide explains the need for better biomarkers to enable earlier detection and more effective treatment of HGSOC, ultimately aiming to improve patient outcomes. In the third slide, we talk about methodology overview. In this slide, it provides an overview of the research methodology. It highlights the data collection process where proteomics information from ovarian tissue and serum samples was gathered. The next step involved in extracting features from this data, focusing on identifying co-dysregulated proteins associated with HGSOC. Finally, machine learning models were also applied to detect the most promising biomarkers from these features. In slide 4, we understand about the biomarker discovery process. The discovery process involved in identifying co-dysregulated proteins, that is CDPs, that show abnormal expression patterns in both ovarian tissues and serum samples. This slide explains how these CDPs were found to be involved in several important biological functions such as blood clotting, fibrin clot formation, extracellular matrix organization, and cell interactions involving integrin. Understanding these processes helps in selecting the most relevant biomarkers. Slide 5 talks about the key biomarkers that were identified. So, the list of key biomarkers identified in the studies are as cluster 1 CDPs, the proteins like EEF1G, MSLN, BCAM, and TAG-LN2 were identified as significant markers. For cluster 2 CDPs, CRISP3 and MMP9 were highlighted. Additionally, other potential markers like PFM1, CRP, EPX, FGG, PDLM1, and SERPIN proteins were identified as promising candidates for HGSOC diagnosis and prognosis. So we understand that these protein biomarkers were identified in this research. In slide 6, we understand validation and performance of our research. The identified biomarkers were validated using independent datasets to ensure their reliability. This slide explains how the biomarkers achieved a high under the curve of 97%, over 97%, indicating strong performance in distinguishing ovarian cancer patients from healthy controls. This slide also notes that these biomarkers outperform traditional clinical markers such as CA125 and HE4 in terms of accuracy. Slide 7 talks about the biological insights. Beyond their diagnostic potential, the identified biomarkers provided new insights into the biological processes associated with HGSOC. In this slide, we understand how biomarkers are linked to the critical pathways like cell adhesion, extracellular matrix, remodeling, and immune response. The research also categorized CDPs into 4 distinct groups based on their expression patterns in both serum and tissue, which further enhances understanding of the disease's pathology. In slide 8, we understand the clinical implications and of research findings. We understand the identified protein panels showing significant promise for improving the early detection and prognosis of HGSOC. The increased diagnostic accuracy offered by these biomarkers could lead to earlier and more targeted treatment strategies, ultimately improving the patient outcomes and survival rates. And in slide 9, we understand the future directions. This slide outlines the next steps of the research, and it emphasizes the need for further validation of new biomarkers through additional clinical studies. The slide also discusses the potential for integrating these biomarkers into routine diagnostic tests for ovarian cancer and explores the possibility of applying these findings to other types of ovarian cancers and related diseases. In slide 10, we understand about the conclusion, and this slide summarizes the key findings of the research and highlights the successful identification of new biomarkers for HGSOC by machine learning techniques. These biomarkers offer a promising tool for improving and diagnosing treatment of the ovarian cancer. The slide includes emphasizing the potential impact of this research on ovarian cancer diagnostics with the hope of significantly enhancing patient care and survival outcomes. Thank you.